In this video, we will discuss about immunosuppression by activation of innervating neuron effect on tumor growth. So here what we are discussing is that whether the immunosuppressions that is caused by this activation of innervating neuron can also affect this tumor growth, and what are the other factors that plays or helps in this tumor development. So in order to do this analysis, what they did is they take this two different tumor models, that is B16F10 and SCC7. So here the results shown for the B16F10, where what it is, they did the RNA-seq analysis of the samples of two different conditions. First one with this chemogenetic inhibition, that is this HM4D-GI, and second one is the control, which does not have the chemogenetic reagent. And they are able to see the genes that are upregulated in the chemogenetic inhibitions, and those are using this gene ontology analysis. The top 10 signaling pathways that are upregulated are shown here. In which is regulation of innate immune response, activation of innate immune response. This indicates that immune response is high in the chemogenetic inhibitions compared to the control case. Afterwards, they did the mass spec analysis of this B16F10 tongue xenograft using the of this tumor derived lymph node to find out what are the different kind of cells that are present within these tumors, and they plotted this they did a Disney plot to find out different populations of the cells that are present, and they did this analysis for 4 different conditions. First one is this wild type, second one is this chemogenetic reagent, third one is by triggering immunosuppressions using this catch, that is enhancing calcium in the intercellular part of the cells, and third one is adding the chemogenetic inhibitions to this catch model, where they want to see whether this chemogenetic inhibition overcomes the tumor suppressions promoted by the catch. And this catch system is basically light driven or optogenetic driven. That is why the lambda is there. And what they are able to see is that in the case of this catch system, SCN10A catch system, they are able to see increase in the population of M2 active macrophages, and also there is an increase in this PD1 CD8 plus T cells, indicating that this system promote immunosuppressions and also the tumor associated macrophages, and that contributes towards this tumor growth, that might have a role towards this tumor growth. Whereas when they use this fourth system, that is when they added the chemogenetic inhibitions to this catch model, they are able to see that there is a decrease in the tumor associated macrophages, and also the dendritic cells, both the conventional dendritic cells 1 and conventional dendritic cells 2 goes up compared to the case where only the catch model is used. And at the same time, the regulatory T cells also goes down, indicating that the immunosuppressive environment goes down whenever this chemogenetic inhibitions is been used or is been combined with this catch. And these things are also true when only the chemogenetic inhibition is done using only this HM4DGI reagent. This in all together indicate that these activations of this innervating neuron can cause this immunosuppressive environment through the increase in this M2-like macrophages. And M2-like macrophages are also known for this tumor promoting macrophages, and they helps in this tumor development, whereas M1-like macrophages are known as tumor suppressing. But there is no change in the M1-like macrophages populations, but the main changes that are found in this tumor associated macrophages, conventional dendritic cells, and this PD1 CD8 plus T cells. This also goes down whenever this chemogenetic inhibition is combined. And this PD1, as we know who are following this channel, that PD1 is one of the PD-1 is one of the common immunosuppressive receptors that suppress the immune response. So this also goes down whenever this chemogenetic reagent is used, along with this catch E by FP. So this in all together indicates that the immunosuppressive environments are being triggered by this activation of innervating neuron, and this immunosuppressive cells are being suppressed whenever this chemogenetic inhibition or chemogenetic reagent is being used along. Chemogenetic reagent is used either along with this catch or individually. So I hope you understood how this immunosuppressive environment affect the tumor growth through the activation of innervating neuron. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.